Welcome to today's MPG Primer. We're excited to be in the big auditorium for today's speaker, Dr. Chrissy Austin-Tse. She here is the analyst team leader for Broad's Center for Mendelian Genomics, where she manages the team of genomic variant analysts focused on identifying causal candidate genes and variants in genomic sequencing data. As a board-certified molecular geneticist, she also signs out clinical lab reports at the Broad Clinical Labs and the MGB Laboratory for Molecular Medicine. I think I've looked at many of her reports myself, so always appreciate them. She's an assistant professor in pathology at MGH, as well as Harvard Medical School, and an associate member here at the Broad. She received her PhD in the BBS program at HMS and her fellowship in clinical molecular genetics at the HMS Genetics Training Program. We are so excited to have her here today to speak about seqr, which I know I use all the time at Boston Children's Hospital, and it's an exceptional platform. So looking forward to learning even more about it today. Dr. Austin-Tse is very happy to take questions during the talk, so feel free to come up and speak at the mic, or if you're online, just chat in the Q&A and I'll voice your question for you. Thank you so much.
Thank you. Thank you for that introduction. All right. So, as was just mentioned, I lead a team at the Center for Mendelian Genomics that's really focused on using genomic analysis to find answers for rare disease patients. So, diving right in, what do we mean by genomic analysis? This term, or genomic sequencing rather, this term encompasses both genome sequencing, looking at all 3 billion base pairs of the human genome, as well as exome sequencing, looking at just the slice that encodes protein, which is still about 20,000 genes. So the overarching story here is that with both of these sequencing methods, we are generating a lot of data. So in an exome, you're going to see about 50 million reads. In a genome, 1 billion reads. And when aligned to a reference sequence, this translates to about 100,000 variants in an exome and millions of variants in a genome. So really, this presents us with an issue of how do we go about finding anything informative in this sea of data. And so the process that I'm going to talk a lot about today is what we call filtering. It's really where we start with the millions of variants that were called in the genome sequence, apply a series of criteria to gradually narrow the scope of variants we would need to look at to hopefully identify a causative variant for our case. A critical first step in doing this filtering process is to take the actual variant calls in your VCF and append them with all of the information that you would ultimately use to filter the variants. This is a process called annotation. So there's a lot of different information that you can tag on to these variants. For example, does the variant impact a gene? If so, which gene? How does it impact that gene? Is it a coding effect? Is it a splice site variant or other non-coding type of variant? What is the zygosity of that change? So is it a homozygous call, heterozygous, et cetera? Quality metrics that give you confidence that this is an accurate call, population frequency data, or variant disease associations from databases like ClinVar, for example. So as I talk through filtering, I like to do it in the context of a case. So let's start. We have a male patient in his teens who had these recurrent oral ulcers. He had a very notable family history, including a father, paternal grandmother, and sibling that also were noted to have those same oral ulcers, and a whole exome was ordered. This is maybe a slightly atypical indication for an exome, but nonetheless, we're starting with an exome here. And if we start with 67,000 variants that were identified from that sequencing test. So how do we start to go about narrowing that scope down? One thing I already mentioned briefly was population frequency data. So this is a really easy kind of, I think, way to think about starting to narrow your scope. The problem here is, of course, that we all carry a lot of variants that are way too common in the general population to be disease-causing. So the example I'm showing here is an A to T change. 60% of the general population may have an A at that position, but if 40% of the general population has a T, that A to T change is way too common to be causative of a rare Mendelian disorder. So an easy first step that we could take would be to filter all of our variant data down to those more rare variants. We use population databases, very notably Nomad, in order to do this. It does bring up the question of what is an appropriate frequency cutoff to apply. And this does depend a little bit on the phenotype you're looking at, how it's inherited, how penetrant is the disorder, so how likely is it that someone with a pathogenic variant in that gene will eventually develop disease. But a very common starting point that is honestly overly permissive, but a generic starting point that a lot of people would use would be a 1% frequency cutoff. So let's use that example for our case. Say we applied this less than 1% minor allele frequency cutoff using Nomad data. We would get from 67,000 variants down to 7,000. So this is already a huge reduction in the scope of variants we need to look at, but clearly we still can't look through 7,000 variants. So where else could we go with this? This is where you start to have a lot of different options for how you can approach filtration. And I like to bucket them into 3 major categories: variant-centric, phenotype-centric, and inheritance-centric. So I'll start with the phenotype-centric approach because I think this is kind of a logical path that a lot of people think of doing first. And really the idea here is that you want to look at all of the variants in genes that you already know to be of interest for your patient's phenotype. So this sounds easy enough. However, it is actually kind of annoyingly hard to define what genes you should look at for any given phenotype. And this is really because there isn't one super comprehensive source of gene disease associations. Part of the problem with building gene lists for analysis is that as soon as you make them, they're essentially out of date because gene discovery is ongoing. So what we do when we're trying to take this approach is actually to assemble gene lists from multiple different sources. So these could include variant databases like the ClinVar database, where clinical labs and researchers can submit any variant that they've identified along with its classification and hopefully phenotype information as well. HGMD, which is a more proprietary database where they've mined the literature and created a structured database of published variants and their associated diseases. We use gene disease reference materials like Gene Reviews and Medline Plus will give you an overview of the very well-established genes for a given disorder. OMIM has more emerging associations between genes and diseases. And is definitely more up to date, however, still falls behind in some respects for, as I said, it's very hard to keep up with the pace of gene discovery. Panel App is a really nice resource, which is kind of a crowdsourcing approach to gene list building that was developed by Genomics England, where experts can go in and kind of nominate genes that are associated with their favorite disorders. GenCC, we like to fondly refer to as the ClinVar, the ClinVar of genes, I should say, where again, research groups and labs can submit any gene disease association pair that they've evaluated and the strength of that association. Other places to look are, unfortunately, the scientific literature. We end up coming back to looking through recent articles to find newer associations with disease. And I've listed the genetic testing registry there as a resource of if there are clinical tests available for your phenotype of interest, it probably makes a lot of sense to ensure that you've captured those genes as well. So the approach here would be to generate your list of genes, and then your subsequent filtering approaches that you might append to that really depend on how long your gene list is. So if you end up with just a few genes, you may decide you want to look at all of the variation in those genes and evaluate it. If you end up with a very large gene list, which will happen for disorders that have a lot of genetic variability, like neurodevelopmental disorders, you probably need to add on some additional filtering metrics there, maybe start with coding variants, et cetera. So let's move on to this inheritance-centric approach. The idea here is really just reviewing variants based on interesting inheritance within families. So there are a lot of ways you can approach this, but just highlighting the 3 major ones that we often see. Considering first on the left here, a de novo case. So this is a family where we have an affected individual in red here, and the parents are not affected, no other affected members of this family. So one powerful approach in this situation would be to look for de novo variants or variants that are found within your affected proband but absent from both parents. We all carry around 1 to 2 de novo variants in our coding regions, so the fact that the variant is de novo does not automatically mean it's pathogenic. But if that de novo variant occurs in a gene that is highly related to your patient's disorder, that's somewhat strong evidence. Moving over to the right here, this is another family where, again, we have an affected individual, an affected child with both parents unaffected. You can also consider a recessive inheritance pattern in this case, where both of the parents actually have a variant in the GJB2 gene, and the affected individual has inherited both of those variants, so has 2 affected copies of the gene. So the filtering approach here is essentially to look for genes that have 2 variants within them, one inherited from each parent. And then lastly, in a dominant disorder, it's great if you have sequencing data from multiple affected individuals, and you can do a nice filtration to look for variants that are shared between the affected individuals as well as absent from the unaffected. All of these, I'm giving a pretty high-level overview. All of these have caveats. So for example, in a dominant disorder scenario, you do have to be careful about excluding all variants that are present in unaffected individuals. For example, if you have a case of reduced penetrance. That's the general approach there. And then the variant-centric approach is essentially asking to look at any variants that are inherently interesting based on properties of the variant, and then go back and decide if they might be relevant to your patient. So the approach here, this is my preferred starting approach. It's essentially deciding on buckets of variants that you think are particularly interesting. So they may be the variants that have been previously reported as pathogenic in a database like ClinVar, or variants that have a severe impact on the protein, like loss of function variants, may be narrowed down to known disease genes, et cetera. So say we took that variant-centric approach on our case example here. We had the 7,000 rare variants. Say we applied this filter of ClinVar pathogenic or likely pathogenic. You can see that instantly gets us down into the realm of 6 variants, and this is totally feasible for us to go in and then manually review. So this manual review process is our next step, and it's something that is referred to as triage in a lot of labs. While the analyst is doing this, they're essentially simultaneously asking themselves a number of questions. They're looking at the gene that the variant falls in and deciding if it has an association with human disease already and the strength of that association. They're deciding if that disease that the gene is associated with is somehow relevant to the patient's phenotype, and also deciding if the variant itself is likely to cause that disease based on its impact to the gene. So this requires the assimilation of a lot of different types of information, and is a very time-consuming process, easily the hardest and longest part of our analysis process. So there are 2 major points I want to make here. One is that your filtering strategy really has to strike a balance between capturing all of those variants that may be the answer for the patient, but also limiting the number of variants that need review, because this is such a time-consuming process. The second is that because there's so much information for the analyst to process here, having a software tool that presents you with a lot of this information that you need to consider has a big impact on your analysis time and your ability to accurately analyze. So just to drive that home, the display of your variant information can really make the difference between what just looks like a missense variant that's not too interesting versus what is probably an explanation for the recurrent oral ulcers in this family. So in our example here, this is a missense variant, but it has been previously reported as pathogenic. It's extremely rare, not in the NOMAD database. You can see that the gene is associated with a mucocutaneous candidiasis, which if you read into it, you will find actually causes recurrent oral ulcers. So you can see that this makes a big difference, and this would immediately stand out to an analyst based on all of these nice annotations that we're displaying here. and also that it was inherited between the affected father and son. So, I presented that as a nice example of finding the answer for this family, but this is actually an extremely limited search. In order for us to have found that variant with our filtration approach, as I just described, it had to have been already detected by another laboratory. That laboratory had to have enough information to classify it as path or likely path, and then they had to make the effort to submit it to ClinVar. So this is not an example of our standard filtration approach. In fact, we usually take a much more comprehensive approach that really incorporates all of the 3 different buckets of search possibilities that you could go down that I just described. So we may look at the previously published variants, but we're also going to look at loss of function or high impact variants that are rare. We're also going to do a gene list-based analysis to see if there's any interesting variants in genes that are known to be associated with that phenotype. And we're also going to look at variants based on their inheritance pattern as well. So you can imagine that as we were considering this more simplistic filtration approach, we could probably do this fairly easily within a spreadsheet. But as you're layering on all these different filtering approaches, it gets more and more complicated to do that. So you really need a software tool to assist you in that process. And this is where seqr comes in for us. So the seqr tool was developed within the Broad Center for Mendelian Genomics to assist with our genomic analysis, but has since expanded in its role to really play a key role in our data sharing and collaboration between teams as well. So before I move on to talk through some of the features of seqr, I just want to highlight our small but mighty developer team for seqr, including Hannah Snow, our technical lead, and Lynn Pice, who is our product owner and here in the audience today. So if you have further questions after the talk, she's a great resource. All right, so seqr is a freely available and web-based platform. So I put the URL at the top of the page here. Anyone can go to seqr.broadinstitute.org and log in. It does require a Terra account, but that's very easy to set up. Once you sign in, you should see a page like this, which will list all of the projects that you have access to. So anyone who logs in will have access to the seqr demo project, which contains a few genome sequencing families for you to play around with, test out the seqr features. You can't break anything in that project. We clear all the notes and tags in it periodically, so that's really there for you to play around with. If you were to click on the name of that project, you would open up the project page here, which has an overview of the number of individuals and families within that project. And if you scroll down to the bottom of that page, you'll see the list of those families. You can click on the family name and it'll take you into the family page. There's a lot of useful information on this page, so I'll spend a little more time here. We have a pedigree to show you the relationships of all the individuals in that family and also lay out who's affected and unaffected. We have a place to track the status of that case, so whether it's still under analysis, if we consider it solved, or we have a strong candidate that we're considering for follow-up. We have the patient-level information, including age. There's a free text field for phenotype information, so you could enter a more narrative description of your patient. But there is also, very importantly, structured phenotype information in the form of HPO terms. So we have on the right here the data that has been loaded for that patient, as well as the date it was loaded. And then up in the top right is where we can start our variant search. So if you click on this variant search here, it will take you into the seqr variant page. All right, or the variant search page, rather. And so I'll just walk you through all of the options here to give you a sense of how customizable these searches are. The first is the inheritance section, which, as the name implies, where you can filter how this variant is inherited throughout your family. So you can set allele counts for each individual member of the family, but we also have preset options here where you can just select recessive or de novo dominant, and it will preset the appropriate allele counts for you. In the pathogenicity section is where you can filter based on whether this variant has been previously reported, so pathogenic or likely pathogenic in ClinVar, or disease-causing in the HDMD database, for example, if you were looking for those previously published variants. And this has a bit of an override to prevent us from over-filtering. So if you select pathogenic and likely pathogenic in ClinVar, as well as later select a frequency of 1%, it will override to force you to look at anything that's pathogenic or likely path in ClinVar up to a 5% cutoff, just so you don't lose variants that are maybe higher frequency but known to be disease-causing. The variant type section here is where you can select your consequence on the genes. So you can go in, and if you wanted to look at loss of function variants, you might select the nonsense, the essential splice site, the frame shift variants, et cetera. There's a lot of annotations, including both coding and non-coding impacts, as well as structural variant impacts. In the frequency section is where you would filter based on your nomad or top med allele frequencies. There's also a section to filter on the frequency in our own call set to get rid of sequencing errors that are inherent to our technology. And then in the location section is where you can filter based on your gene list. So you can either manually enter a gene list into this genes intervals box here, or a really nice feature that was more recently added was we have automatically ported in all of the available panel app gene lists. So in this header menu here, you can just type ahead your phenotype of interest. It will pull up a list of all of the relevant gene lists that are available within seqr, including all the panel app gene lists that are available, and auto-populate those genes for you. And then finally is our call quality section, where you can filter based on genotype quality, allele balance, and hard filters in VQSR. Okay, so I've shown you how customizable that search is. Some people find that a little intimidating. How do I know how to set up the appropriate search? We have some guidance there as well. We have a set of standard searches that we routinely apply to our own cases, and we've saved those as saved searches within seqr. So that can be found within this header menu where it says select to save search here, just outlining them here. Essentially, we have search options for de novo and dominant and recessive, and each of those is broken down into restrictive and permissive options. In general, the restrictive searches are more highlighting the coding regions and high-quality variants, whereas the permissive searches expand out to more non-coding annotations and relax our quality filters a bit. I didn't highlight very well the actual phenotype of the next example case we're going to work through, so to go to that quickly. Let's look at this case now where we have some phenotype terms down at the bottom here, abnormality of the kidney, which is a polycystic kidney found in utero and multicystic kidney dysplasia. Based on this pedigree here, we have an affected individual with unaffected parents. We might start with a dominant search approach. So we'll start with our de novo dominant restrictive. We'll select that in our menu here, and you click the search button at the bottom under all these search parameters, and that will bring up the variant results within seqr. So one thing I've highlighted here already is that this returned only one variant. It's very nice when you have a trio. You will often see very few variants returned by these de novo dominant searches. You will see more, however, if you are doing a proband-only sequencing approach. And we'll talk a little bit about how you can handle that. But I first want to just quickly go through all of the information that seqr is presenting to us so we can evaluate each of the candidates returned by the searches. So let's start on the left here with the gene. Information is at the far left. You can see that this variant is falling within the BIC1 gene. There are some high-level tags that show you loss of function constraint for this gene, as well as haploinsufficiency. We can see the gene is present within the GenCC database. If you hover over that tag, you'll get information about what diseases it's been associated with, as well as the strength of those associations. And we also are displaying information from OMIM, so we can see that this is a gene that has, at least in OMIM, been associated with a renal cystic dysplasia, which is interesting already for the phenotype of our patient. In the middle here, we have our variant info, so you can see the C dot and P dot consequences of this variant. You can see how it is inherited in this family, so we can see that it is present in our affected individual, and all of the unaffected individuals in this family are negative for the variant. Importantly, if you don't have a system of naming your subjects that you can automatically tell their relationships based on how they're named, you can hover over the family number here and it will show the pedigree again for you, which is nice. In the middle, also, we have our in silico predictors for this missense change. We have a number of them shown automatically. You can click on Show More to have even more options, and we are constantly reevaluating which predictors to add to seqr, so you can always shoot us a note if you have a favorite one that you really think we should add. seqr has a really nice feature in that the read data is automatically built in. So if you were to click on this Show Reads link, you would see a IGV screenshot show up underneath. And you can review the location of your variant and see if you think it's real, which is nice. And then at the far right, we have our call set frequency data, Nomad, as well as TopMed, and our own call set. I mentioned when we do a proband-only search, you're going to get a lot more than just one variant from searches like this. It's very nice in seqr. We have the ability to sort our search results by a number of different metrics. So you can see the example here is sorted by OMIM, so it will put any genes that have an OMIM disease association towards the top. You can also sort by pathogenicity, so you have all your ClinVar path and likely path variants at the top, or things like your in silico predictors, so you can have the most damaging rebel predicted variants at the top, et cetera. And there are a number of helpful links as well. So say this was a recessive disease gene and you were really interested in this variant, but you didn't find a second hit in your immediate search results, you could click on this gene search link and it will automatically generate a search for other variants less than 10% frequency within that same gene. The seqr search links under the variant will allow you to search the entire seqr database to see if there are any other individuals within the database with that same variant. You can do a Google search to try and find more information in that variant. So there's a lot of resources here. How are we doing for time? All right. So again, for our example case here, as a reminder, you can also hover over this pedigree symbol at the bottom. It will remind you of what your HBO terms are. I have borderline dementia, I feel like, so I forget instantly, and so that is a really helpful feature to me. I end up looking at that a lot. So for this case, we have an abnormality of the kidney, our multicystic kidney dysplasia. I would say I'd be pretty interested to find a de novo variant in a gene that's already been associated with a renal cystic dysplasia. So what we can do to try and figure out, did we find the answer for this case, would be maybe to look a little further into the gene. If you were to click on the gene symbol here, it would bring up this pop-up window with all of our gene level information. Some of it's already displayed on the variant level, but it's a lot more detail here. So there are links out to other resources, for example, mice databases to see if there are any knockout mouse models already for that gene. But what I'm going to highlight here actually is that in the GenCC database, we saw there was a submission. It actually turns out to be a limited association submission, which suggests there's not so much evidence supporting this association out there. That was evaluated by one lab, so obviously we'd have to evaluate ourselves. But this might give us a little pause, and we might want to generate a little more information about this gene. We have a small screenshot at the bottom because you have to scroll further down to see it. But we also have GTEx information in there so you can see if your gene is expressed in relevant tissues. So let's say we are interested in this gene, But really, we'd need more evidence to decide it was the answer for this case, given there seems to be limited evidence supporting the gene-disease association. So the next step you really would want to take to build information and evidence for this gene would be to confirm or maybe find other patients that have a variant of interest in that gene and determine if they have a similar phenotype to your patient. So when you're working with rare disease patients, you can imagine that this is actually pretty challenging to do. There are not a lot of people out there with variants in this gene if it actually is disease-causing. And so this is a problem that actually the Matchmaker Exchange database was built to answer. So this is a federated network of a few different resources that we're trying to address this problem by creating a place where you can submit a gene of interest and find other patients with that same gene. There are a number of connected nodes within the Matchmaker network, such that if you submit a gene or variant to any of those nodes, you can find information from any of the other nodes, which is great. And seqr itself is a node within Matchmaker Exchange, which has really helped us out. So within seqr, we can now submit a variant of interest in one of our cases to Matchmaker Exchange directly through seqr by clicking the Matchmaker link, which is found on the family page. seqr has really facilitated this process for us because it's basically a matter of checking a box. It will provide you with a list of all of the variants that you have tagged as potentially interesting in your case, and you just check the box of the variant you want to submit, as well as the HPO terms that are already associated with that case, and submit it directly to Matchmaker. So that's a very easy process. seqr also facilitates the follow-up. So once you submit to Matchmaker, you get a report back of all the potential matches for your gene from all of the other Matchmaker nodes. So seqr displays that information almost instantaneously after submitting to Matchmaker, and you can see an example here where we've actually matched with Gregor Mendel himself. Thanks to Lynn for that example. Who also has submitted a variant in the BIC1 gene and has a phenotype of renal dysplasia. So that might be very exciting for this case. The next step you would take after seeing all the matches you get from Matchmaker would be, of course, to reach out to Gregor Mendel and decide if he maybe has more phenotypic info to confirm that that is really a match for your patient. Or maybe he's putting together a cohort and you want to talk about if we can get our patient into their paper. So this is a process that is usually just, you get an email back from Matchmaker Exchange. You have to reach out to all of your potential matches for further info. But we have set it up so seqr can really facilitate that process as well. So you can see there's a link here to contact the host. Once you click on that, seqr actually is auto-generating an email for us, which has the information that we would typically send in these types of communications, including the variant information and some of the information about the phenotype of our patient. This is, of course, editable, so you can change it however you want. But we've found this really helps us in doing a lot of these connections quickly. So you can send that email directly from seqr. As you build up more and more candidates and you have more and more submissions to Matchmaker, you'll find that managing all of these Matchmaker communications actually gets to be a job in and of itself. So we have designed seqr to facilitate that process as well. Once you've sent the email within seqr, this tag that says not contacted will update, so you have a record that you've contacted this person already. There's also a free text field where you can go in and write notes about any of the information that was exchanged. So it's a nice place to kind of collate all of that information together. So in summary, for this case, I would say that our BIC1 gene is a very promising candidate. We actually did submit it to Matchmaker, and though we did not match with Gregor Mendel, we have found other interesting matches, and we do think this is a likely answer for our case. So hopefully, in the process of this talk, I have given you a sense that seqr is a very useful resource for genomic analysis. Just to drive that point home, some basic stats from our center that we've now analyzed in seqr over 46,000 cases. and found answers for over 4,000 families. We've done a lot of Matchmaker Exchange submissions, so over 1,000 genes at this point have been submitted to Matchmaker through seqr, and through this we've made over 300 novel gene discoveries. So we really think seqr is a great platform, obviously. We built it, but hopefully you think so as well. So if after watching this you're interested, you'd like to try and use seqr with maybe some of your own data, how would you go about that? We have recently made seqr available on Anvil. So anyone who has data on Anvil can port it into seqr. We have a little YouTube video describing this process to hopefully lay it out quickly, but briefly here, what you'll need to do first is create your Anvil account. It only takes a minute, and you can get straight into seqr, and you'll be able to see that seqr demo project, as I said. To have your own data, you'll need to set up an Anvil workspace, which will incur some cloud costs, and you'll have to set up the permissions appropriately. But seqr does require a joint called VCF, so you will need a GATK joint called VCF. You can either bring your own joint called VCF or generate one with Terra workflows. And then there is a link at the top of the workspace to request data loading from Anvil to seqr. I do want to note that this does take a little bit of time, so give us 2 to 7 days to actually load that data into seqr. At that point, you can upload your project metadata, like your HPO terms, other relevant case info, and you can go ahead with your analysis. If you want some more information about seqr, we do have a publication out in 2022. We have, as I mentioned, that one seqr video on Anvil loading. We actually have a whole YouTube playlist on seqr features that can be found on the Broad YouTube. There's an FAQ page I've listed there, and if all else fails, we also have a general seqr email address that you can reach out to with questions. So I have to thank, really, it has been a collaborative effort of everyone within the Center for Mendelian Genomics as seqr has evolved over time, with special thanks to our fearless leaders, Anne and Heidi, who've really spearheaded not only evolving seqr into what it is today, but making it more broadly available to other researchers as well. And thanks to you all for listening. So I'm happy to take questions if there are any.
Great, thank you so much for that excellent talk. Feel free to type in the Q&A if you're at home, or come up to the mics here, they're both on. I had a couple of questions.
Oh, yeah.
When you were looking at the in-cohort allele frequency, just the thought of someone who brings their own data through Anvil, do you have a recommended minimum size then for a jointly called VCF? Like, should you throw in some more individuals so that you can have an in-cohort allele frequency to filter on?
So
or?
previously, that in-cohort frequency was highly dependent on your own data set. Lynn, correct me if I'm wrong on this, because this has evolved recently, but now that data is available for all of seqr's call set. Is that correct? Yeah.
Anvil represents all that data.
Oh, perfect. So the in-cohort is actually all available VCFs that have been loaded. That's exciting. Is there a way to then be sure that the variant that's in cohort is not the same one that might be in Nomad? You know, like double counting some individuals who may have, I guess that would be unlikely. Perhaps
If that the same person is in our cohort and also in Nomad. So we haven't loaded any Nomad data into seqr, if that helps. But yeah, it would be hopefully a rare instance. Yeah.
We every now and then find individuals in our research cohort who are so generous with their participation in science that they've enrolled in other cohorts also.
That's tough. I should point out that I mentioned that seqr search link will bring up that variant and anyone else in the cohort. So you will actually see a display, even if that individual is not within your data set. It's a very rudimentary display, I think, of that information at this point. So it's not identifying that individual, obviously, but you might be able to see some high-level HPO terms that might give you a clue. Yeah.
Sorry, just making sure there weren't any Q&A questions. And I guess another question for this is with the seqr instances, they've been implemented at some of our local hospitals, obviously, to great effect. Do you feel like this is going to branch out into other medical systems, or are you going to have people mostly enter via Anvil?
Yeah, it's a great question. So, it depends on the needs. I think in clinical systems, it generally makes more sense to have a local instance due to all the regulatory issues along with that. However, there is a lot of engineering needs needed to support your own local instance. So I think that's a big lift for some groups. So it really depends on the resources available to set it up. I don't know, Lynn, if you have stats off the top of your head versus Anvil users. We certainly have the vast amount of more of Anvil users versus local.
We like to encourage people to use it on Anvil because you just have more features available. We like to encourage people to use seqr and Anvil because, as Chrissy mentioned, there's low burden of maintaining your own version of seqr. And a lot of features, which one here that Chrissy is showing is the variant lookup, is you can have access to all of this information. So if you have your own local instance, it's not connected, it's isolated from this. As well as when we launch newer features, it's available first on the Anvil version of it. And then it takes a little time to put out the documentation so you can update your local version. So bottom line, unless you have strict restrictions on if your data cannot leave the country, like some countries like Canada and Scotland cannot have data in the cloud, then it makes more sense to have it in your own version. But otherwise, we really encourage people to use it on Anvil.
Good point.
That sounds great. Thank you. Perfect. Well, thank you so much for a lovely talk today. There's a little bit of time to get a bit more coffee and breakfast, and then we'll start again at 9:30. Thanks.